Scientific American. Most people are familiar, it's kind of cutting out, sorry. Well, as I said, most people are familiar with the basic functions of RNA, and they like transcription, translation, and how it makes proteins. This is the basic like functions that we all know. And it uses like the little sequences here of codons to make amino acids that makes proteins in our body. And normally people just assume that RNA just is kind of like it's used by DNA and it's used by proteins to help make proteins. But it's a little bit more complex than that. What scientists have learned many new things about RNA in the late 20th century. I don't know why it's doing that, but just work with me. RNA, and they found out that RNA is not only controlled by DNA and proteins, but it also helps control the DNA and proteins. This is just a picture of the differences between RNA and DNA. So scientists have noticed that the old methods of treating people with diseases and just medicines, any medicines from like aspirin and Advil to like more complex medicines, normally they target proteins, but you see here it's a little example. A lot of times proteins are really big and complex and their binding site is found somewhere in the middle, and a bigger protein drug can't necessarily reach those binding sites. And so they're looking for ways to make treatment smaller in order to be able to treat diseases that were considered undruggable. There are 3 new exciting ways that you're able to treat patients utilizing the knowledge of RNA, and these ones are siRNA, which stands for small interfering RNA, microRNA, and the CRISPR system. And these are 3 ways that I'm going to discuss about how you can treat patients. These 2, the small interfering RNA and microRNA, go under the category of RNAi, which is interference, and CRISPR system is on its own. Small interfering RNA are used to disrupt any unwanted mRNA, messenger RNA, which goes to the ribosome. And investigators in experiments have seen that this siRNA with the protein attached to it will just break the strand of mRNA and not allow it to be translated, and they just essentially disrupt the chain of amino acids and block the protein that is affected. MicroRNA can be used to customize the RNA strand and alter specific proteins that you want. As you can see in this one, there's a microRNA with a protein complex attached to it, and as it goes to the ribosome, that mRNA can't go through because the microRNA blocks it. It's essentially what I read it's like a roadblock for the mRNA, and that it doesn't allow it to keep going through, and the protein that causes the disease is stopped and is not able to be translated. This CRISPR system is what I think is one of the most intriguing out of the 3. It stands for Clustered Regularly Interfaced Short Palindromic Repeats, and what it is a strand of DNA that's really, they say it's strange that it repeats so much, and it's just very repetitive strand, and it is found in bacteria and archaea cells, but they harvest them and are able to use them for their benefit with RNA. The CRISPR cells are also they use a what they call a Cas protein, or CAS, and it stands for the CRISPR associated proteins, or Cas proteins associated. And they help form a microbial defense against proteins that cause harm. And what they do is there is a certain strand that is attached to the protein complex that will match up with a specific spot in the DNA, and they make a break in the DNA. And so you can target any spot that you want on the DNA and just break it very, very specifically. CRISPR method has only started with just recently in the last couple years with animal tissue, and scientists are starting to create their own target strands. So you can read the DNA and what is in the body, and then scientists can create a matching strand to target to the gene where they want it. And also another good thing about the CRISPR method is that right out of the box, it's able to be used because it's something that's very natural, like a protein complex would naturally cut, but they're just figuring out how to use it and cut it in the spaces they want it to. And people have asked what makes RNA so desirable over just the standard use of proteins. One is that they're very small, and you can use them at targeting a lot more types of proteins. But the other thing is that an RNA is a two-dimensional sequence, whereas a protein has sequence and structure, and unless it's in the correct structure, it won't work. But RNA, all you need is the right order of amino acids, or the right order of nucleotides, and it works. So as you can see, which you would rather do a math problem on? Would you rather do it on this third grade graph or a more complex three-dimensional graph? I think all of us would say two-dimensional is better. Some areas for further RNA research include incomplete RNAs, which are used to modify the shape of DNA and make some portions of the DNA twisted while it's being translated so that it just isn't able to be read. So that's even going one step further up the ladder and just going straight to the DNA and just kind of hiding some parts of it. The CRISPR system is only, once again, it's only been used in human tissue for 2 years. And so just finding better ways to specifically target spots on the genes. And then implementing theory into practice is one of the things that is, because this is very new technology, it's only been around for around 20 years. And ideas are being implemented in the labs, but not very much have been implemented in actual patients. Over 200 studies have been done, and that's just in the last 20 years. Studies done on Ebola virus and hepatitis C are the popular ones, because hepatitis C is a virus that, in the last 25 years, has become very, very, very harmful to people, whereas 25 years ago, they didn't even know that it really was around. And then it's also, there's a ton of money going into it, and so the funding is there, but they just need people to use their mind power and figure out how to get it available to patients. That's it for the RNA revolution.
Any question?
Joe.
Oh, so you said that can also be used to treat Ebola. So with the recent Ebola outbreak, are they doing more research towards using that method?
Yeah, that's one of the reasons why they're doing it, is because there's Ebola the outbreak, and also there's fear of it being used in terrorist attacks. That was a little side note on the article, was that it's one of the, Ebola is a virus that's very well suited for bioterrorism.
So how can RNA-based treatments treat people with the Ebola virus?
What would happen is the Ebola virus would be affecting proteins in your body, and the RNA treatment would go in and kind of above the level where the proteins are formed and stop those proteins from even being made that way, and just make them normal.
Thank you. So we have another presentation, but let me ask you, can you, because we started a little late today, can you stay here about 5 minutes after our regular time, or everybody should leave at 10:30, or 5 minutes? 5 minutes.
I have a class that's really far away.
Oh, far away. Shout, it's your turn, right? So you have another, you have a class. Okay, then maybe We can postpone her presentation on Monday. You can turn in on Monday your PowerPoint. You can just post your PowerPoint PPT file on the Blackboard here.